Hello friends, welcome to BMH Learning. This video is about Luria Delbruck experiment. Before we begin our discussion, let's remind about Lamarckism and Darwinism. The basic idea in Lamarckism is that the new features are the result of the environmental conditions. Or we can say the environment triggers the development of new features. Theory of Darwinism states that the different organisms arise and develop through the natural selection of small inherited variations. So that means the new features are already present and they are chosen on the basis of their survivability through the process of natural selection. Now, the Luria-Delbruck experiment cleared the concept about these two, because only one of these two can be true. So the experiment actually proved the theory of Darwinism to be true and discarded the idea of Lamarckism. Now let's talk about the experimental idea. It's basically based on mutations. Luria and Delbruck, these are the scientists who conducted this experiment. So they used E. coli bacteria and a virus that is basically a T1 bacteriophage which lyses the bacteria or which eats the bacteria. Now in this experiment, two types of bacteria are observed. First is the T1 bacteriophage sensitive bacteria. So these bacteria die in the presence of virus. The second is T1 bacteriophage resistant. So these bacteria are not susceptible to phage and hence are resistant and will grow. So a pure culture of bacteria is taken. Now there are 2 points to be noted. First, we don't know anything about the resistant capacity of this bacteria to the bacteriophage. Or we can say to what extent the bacteria is susceptible to the phage. But we do know one thing, that if the bacteria is in the wild form, in that unmutated form, the phage will surely eat up the bacteria, because that's what these phages do. Now let's see, will the phage lyse it or not? So there are two scenarios. Scenario number 1. The E. coli from the culture is transferred to a petri dish. Now, this petri dish actually harbors the T1 bacteriophage. So the bacteria should not grow on this plate. So normally, the bacteriophage will eat up the E. coli. That's just an idea, okay? So this is the case of T1 sensitive bacteria. Scenario number 2. Again we transfer the E. coli to a plate carrying bacteriophage. But this time the bacteria actually survives. It grows. So in this scenario, the bacteria are mutated. And because of this mutation, they have become resistant to the action of bacteriophage. So this is the case of T1 resistant E. coli. So we are gonna observe the colonies here. The number of colonies may vary in different plates. Now there are two possibilities. First possibility, the mutation was pre-originated, or in other words, it was already present in the bacteria before its exposure to the bacteriophage. Second possibility, the exposure of the bacteria to the bacteriophage resulted in mutation, or we can say the bacteriophage somehow triggered the mutation in E. coli. So we have two hypotheses here. The first states that the mutations were already in the bacteria, that's why some of the bacterial colonies became resistant. So the resistance is varying in different plates. In some plates, you are observing more colonies than others. So the mutation is happening randomly. That's very important. So random mutation is there. Second hypothesis states that mutation can never happen randomly. It only occurred once bacteria came into contact with bacteriophage. Now, don't you sense something here? The second hypothesis or the scenario is actually pointing towards Lamarckism. Remember, in the beginning we discussed that the new features are the result of the environmental conditions, or we can say the environment triggers the development of new features. This is Lamarckism. And that's what's happening in the second scenario. The phage, which is representing environmental condition, is causing the mutation in the bacteria because they need to survive, and hence they try to mutate themselves. Now the first scenario is actually representing Darwinism because some of these bacteria were already mutated. So we are finding here two ideas, and the first case is actually justifying the Darwinism because not all the bacteria are resistant as the growth is varying in different plates. If it was to be entirely wild type bacteria in the first place, then the exposure of the old bacteria to the bacteriophage must have mutated entire population of the bacteria, and this would mean all the bacteria should have become resistant. And we see that's not happening here because the different degree of growth is seen in all plates. So the data we are getting here is backing Darwinism and not the Lamarckism. So the conclusion is Lamarckism was rejected. This was all. Thanks for watching.